we'll hear insights from our panelists as decision makers in biomarker partnering, discussing what they're looking for in academic innovations before deciding to partner or collaborate. And then finally, we'll reflect on the key lessons that they've learned from their own experiences bringing biomarkers from lab to clinical and commercial success. So with that, let's dive in, guys. So first, for some basic strategy, from your experience, who did you find to be the key stakeholders in getting a biomarker diagnostic test to market, and how do their priorities differ? Who wants to kick us off, Dan?
Sure. So I think in my case, we were really led by our angel investor who also acted as our CEO, and he kind of got us started in terms of funding. And two of our co-founders were clinical oncologists, so they could kind of represent the voice of the customer and the need. But I think that's really the most important thing is to almost think of yourself like an anthropologist and deeply embed yourself with your target customer, study them, figure out where their pain points are, and how is your technology or biomarker going to create value for them. I think that having that defined is really the most important step to get started.
Yeah, adding to that, it for us when we founded Previse, we had these epigenetic assays that we knew worked in proof of concept case control studies, and we did this course called the I-Core course, which is put on by the NIH. Went out and we did this customer discovery program. Started talking to different stakeholders. And to Dan's point, we spoke to GI physicians, we spoke to like some hospital administrators, started to get an idea that, okay, it seems like there's a problem and the solution we're developing seems like it could be a viable solution, but there were so many questions that we had. We started using this term, at least in my background, was familiar with analytical and clinical validation, but we started hearing this term like clinical utility thrown around. And hey, if you ever want your test to be paid for or reimbursed, you're going to need to demonstrate clinical utility. And they talked about outcomes data and how might this test impact decision making within the clinic. And then we started learning about different questions about the market and how's this actually fit in at the clinic? Who's going to be the person identifying these patients? Who's going to be ordering the test? How's that process going to look? We heard about laboratory developed test versus FDA, all these different kind of what developed into an elephant of a seeming problem. And where do we take the first bite? And speaking to the stakeholders that we engaged with, we started reaching out to a lot of people through panels such as this, or seeing names of different founders that came out of the institution or experts in the space. And I started reaching out via LinkedIn a whole lot and just messaging people and looking for anybody I could talk to who had experience and maybe some wisdom. So I guess for me, some of the most vital, there's a lot of wonderful resources out there, but some of the most vital stakeholders I've found are other founders or other experts in the space who are willing to share a little bit of time and kind of talk things through, and for me, kind of turn it into what I call Barney style, simplify it, so I can start kind of more so understanding the landscape and the right questions to ask.
Marika, you're muted. I see you're trying to speak.
Sorry, I clicked twice. No, yeah, I think I can nicely add to this because in our experience, we also started engaging with guideline committees and payers earlier on in the development, just to make sure that by the end of it, we would have a dossier with data that would add value to them too. So they are at the very end of the process, but then engaging with them early on ensures your product market fit is there. Also, it's valuable for them to write it in the clinical guidelines or to make sure that your test is covered.
I guess speaking to that, who are some of the people that are on those committees, if you could share, that are providing that insight?
I think in general, these are clinicians in the field. So they have a background in the disease area and they are either key opinion leaders or close to key opinion leaders. And for example, throughout your clinical roadmap where you do several studies, you could engage with them, setting up these studies, engaging with their institutes to let them gain experience and knowledge on your product. It will not ensure that you are in the clinical guidelines, but that experience will at least make them more knowledgeable about your product.
Dan, go ahead.
Yeah, I was just going to add, especially in the diagnostic space, you often have kind of three key stakeholders in a way. You've got the patient, actually taking the test. You've got the clinician, who's the one kind of really ordering the test, and you've got this third party that's paying for the test. And everybody's going to, and maybe a fourth stakeholder would be regulatory, right? Depending on your strategy and where your test is performed. So everybody wants something different, right? Everybody has a different perspective. And so I would encourage you all to not be shy. People love to give feedback and give input. And, even at an early stage, you don't want your prototype to be so fancy and polished that they hold back. That's actually the biggest problem when you're early on and you want to vet an idea with a couple different people in different areas, is that they'll see that you've put a lot of like polish into it, a lot of effort, like you're really trying. And so they'll feel bad that it's terrible and they won't want to tell you the truth, right? And so don't, just like draw in crayon, but like sketch out the ideas and just get their straight feedback and get it from a few different people in those categories that we talked about, right? The regulators, the payers, the clinicians, and then the patients.
And one of the, if you don't mind, one thing I'd like to add to that is a really good point, is one of the things I've come to learn is that it's never too early to reach out to the different stakeholders or to different people. I know I remember early on when we were thinking about fundraising and man, how are we going to go about this, and looking at like SBIRs, STTRs, angels, and trying to refine like the perfect pitch deck, or trying to refine like these things to present it as if we were prepared to go for it. And when in reality, so many times we weren't quite ready for that step. And what I quickly learned is that the best time to meet someone is early on in the process. It's when I'm the most moldable. It's before I've put in a ton of work heading down some avenue. And we've been really fortunate that a lot of people we've reached out to, regardless of the area, whether it's regulatory, whether it's science, CLIA, payer-wise, most people are pretty approachable and are willing to help share their thoughts and their experience. So just adding to that piece, that at least in my opinion and experience, the earlier the better to reach out. And it's okay if you don't really know much about that area. In general, especially from the founder side, people don't expect you to know all of the different areas. So it's okay to walk in and say, hey, this is new to me. I've got this cool science that we think can really help this patient population or these physicians, and let the conversation start from there.
I guess, so since we know who some of the key stakeholders are who you'll need to appease, could you then maybe walk us through sort of your key steps and milestones for biomarker and diagnostic technologies to get them through commercialization, and what should be your main focus early on in your commercialization strategy? Dan, I don't know if you want to talk about your experience at Stanford.
Honestly, I would let Daniel start first. I feel like he has probably the better perspective of taking something all the way to market.
Yeah, so when we started off with our EsoPredict test, we had these, there's some background clinical validation studies using different sets of biomarkers and kind of going through biomarker discovery and look, we can differentiate, this group from I'll call it the experimental versus the control, and it's performed with this area under the curve, and each new paper kind of had some combination of different markers. I think it's funny, we started up a lab outside of the academic center, And we said, okay, well, let's just try to run these methylation-specific PCR assays. Let's try to run these genes. And I remember nothing worked. It was like, this is a test that uses formalin-fixed, paraffin-embedded samples. It's like from the get-go, DNA extraction. For some reason, it was like we just couldn't replicate, even following the same protocols. And one of the things that I really learned through this process is, one, going through each step and troubleshooting each step, but you're going from, we had to kind of change our perspective and shift our mindset and look at it at going, okay, we're moving from kind of novel, proof of concept or type studies in this playground of a research lab that we were in, to now trying to transform this into a consistently performing product, with no assay failures or very low assay failures that can go through each step consistently, and that we can figure out how we measure this and measure what those acceptable kind of boundaries are for this test. And that was quite an uncomfortable process to go through. I mean, replicating science is tough. And now trying to go and take, something that is usually a novel science, whether it's with novel biomarkers or methods, and now trying to take it and turn it into something that no matter what patient or physician uses it in whatever different cohort throughout the United States or beyond, that, and is processed in whatever way, whether it's FFPE samples and how that pathology group, processes them, that once it gets into our hands in our lab, that it performs consistently. So for us, before it got to kind of independent clinical validation studies, it became a question of, okay, how do we optimize each step of this and analytically validate it? And then it moved to the clinical validation, and then, okay, how's this actually work in the clinic? But for us, it was funny. I did not expect us to be spending so much time even digging into DNA extraction and all of the numerous methods. It would be such it'd be probably such a low impact study to publish, but it's like going through all the different DNA extraction methods. And okay, even though we use this in academia, we're going to modify this. And what influence might these pre-analytical steps even have on the downstream assay, and taking it one step at a time like that. And it was slow and it was rigorous, but it was also really rewarding watching this transform from a research lab assay into a product that does consistently perform.
Yeah, one thing I would add to that is that I thought of, Daniel, is generally you want a way for people to try it, whatever it is, right? Like if it's a biomarker panel, if it's like an assay, a test, prototype, you want a way for people to try it. So like it went in my experience with the CapSeq technology, we had this company called Cap Medical. One of the first things that we did is we rented like a single lab bench in like an incubator space, right? And then we had various potential partners or, people who are looking at us for M&A activity. And of course, they all want to send their blinded samples to you to process, right? And so then it was actually me at the lab bench, like processing the blinded samples because we were like a 4-person company. But you need a way for people to try it, because like in what I do now, evaluating other people's biomarkers and assays and things, that can be a real barrier, at least in how I operate currently. Is great, you have an academic paper published and you can show me the data from that, but like I want to try it on my samples. I've got like a blinded sample set. I've tested other methods on those same samples, and so I want to benchmark you to really measure the value to me that your technology or biomarker can provide. So if you don't have like that single lab bench in an incubator space, there's that way for me to try it, that can be a barrier.
So I guess standardizing, validating, and then before even doing any clinical validation, making a way to validate it analytically. I guess another question along those same lines is, those are sort of some of the key steps and milestones from a technical perspective, and then from a market perspective, what are some of sort of the key steps and milestones? Marike, I know you have a lot of different products in your company's pipelines. How do you think about that?
Yeah, I think one of the most important steps in here is to do clinician interviews. So if you want to make sure that your product addresses an unmet need, and it was already alluded to, but you need to really understand where they're going through and where their pain points are. And for some of our technologies, even before in-licensing them, we already did 30 to 40 clinician interviews. And important is also that not all clinicians, multiple different clinicians can be involved at the same patient in the same pathway. So for example, we have a skin cancer product, you have the dermatologist, you have the surgeon oncologist, and you have the medical oncologist. And these three are all involved in basically using our test, but they have different perspectives and different reasons to do or to refer a patient for testing or not. And it sometimes seems so easily, but then through doing all these interviews, you really see the nuances. And I think if you want to have commercial success, you really need to understand where this product fits and how it fits. And I would say if you step outside of the development of the actual assay, doing these type of interviews and really understanding the market is a very vital step.
Yeah, I couldn't agree with that more. The pull has to come from the customer, from the clinical need, from the need of the customer. As a former inventor myself, we tend to think that that's the hard part, that's the good part, that's the part that matters, is like how cool the technology is, and then that can lead one down a mindset of like what I call technology-driven marketing, where it's like, oh no, it's not selling because the customer just doesn't understand the product, or like they don't know how to use it properly. They don't know they need it yet, or what, we need to educate them. Like, stay away from that line of thinking and go to the customers first and figure out what needs they have, like Marika was saying. I think that is going to be very important for your success.
Yeah, we started our company off and we had a different name, and we were going to launch with a different early detection test. Like we had plans, and as we were out talking to different GI physicians and foregut surgeons, we found ourselves trying to convince them why that first product was like the way of the future, and this is like it was us kind of pushing it, and I think luckily for the success of our business, we ended up pivoting, and where we landed, it wasn't doing an FFPE prognostic assay. It wasn't as exciting in our minds as what we were initially launching with, but we learned where the pain points were for the physicians. We learned what fit within their workflow better, and what benefited them, both for the points of hopefully improving outcomes or quality of life for patients, but also their business, looking at procedures and how's this going to influence, how often we do these surveillance procedures or treatments. And so really great point bringing up, getting out there first before putting in all that technical work, and really building an understanding, not just trying to push and convince people that, hey, this is great, but building an understanding of how this will fit in, or even if this will fit in, or do we need to step back and think it through a little bit more.
Yeah, you mentioned prognostic application. I don't know, Iris, you think this is a good opportunity for us to talk about like the different diagnostic applications? Because I think there are very different paths to market, or kind of funding mechanisms for them, right? So, I think of screening or prognostic as being particularly challenging, because usually you have to, self-fund, you have to raise money from venture capitalists. you're not going to get like a pharma partner who's going to sponsor you and pay for your whole R&D and the validation of your product and take you through a trial and everything. Whereas if you have a companion diagnostic test or biomarker application, then you can go the pharma route, and it's your experience might be very different, where they will, if you've got a biomarker that identifies a subset of patients who respond favorably to their drug, and that is the gating factor for them getting that multi-billion-dollar drug approved, they will generally pay for your research and development. They'll provide the samples. They'll take you through a clinical trial, and at the end you'll have an approved diagnostic device for identifying patients with that particular biomarker who will respond. So that's totally different than if you go to a pharma company and you say, hey, I'm making a cancer screening test, right? We're going to catch cancer early. They'd say, oh, that, you have our emotional support, right? Is what they would say, right? But you need to make a business case then, and you need to go to venture capitalists. You need to raise money based on, how you're going to get reimbursement, how you're going to get regulatory approval, et cetera. So I think it's important to think about which diagnostic application your biomarker or test fits into, and then what are the different funding mechanisms for path to market.
Okay, yeah, and I guess an additional one to that is potentially if you're not starting your own company or working with a pharma for a CDX or something, partnering with an existing diagnostics company to see if it's something that they would potentially add to their own portfolio and commercialize themselves. And I guess that leads us into our next section where I'd love to pick your brains as decision makers for whether or not to add things to your portfolio, and sort of what you're keeping in mind as you're evaluating early stage biomarker-based technologies and whether you'd like to partner on them. So Dan and Marika, I know that you both do a lot of search and evaluation in your roles. So as you are evaluating novel and early stage biomarker technologies, especially those from academia, what are you sort of looking out for? What are the critical factors, and what are the facets of your due diligence?
Yeah, so we do evaluation of the technology on 4 pillars. We look at the technology that's using to get to an outcome. So for example, if you have gene expression, if it's on a PCR platform, if it's on a microarray, if it's on next-gen sequencing. We look for the commercial opportunity. If it's really a niche from a niche or a subgroup of a subgroup, it's getting very small to be commercially attractive. We look at the IP. If there is already existing IP that would prevent you from intellectual property, that would prevent you from commercializing the assay, yeah, you don't have a lot of room to play, basically. But I would say that the clinical aspect is the pillar that is most interesting, because it's the most interesting. And if I see the technologies we reject, it's usually on this clinical aspect. So either it doesn't address an unmet need, like previously stated. Sorry, I lost my train of thought. Well, but I think that the encouraging part is also that addressing the unmet need is relatively easy to improve early on. So, and I actually wrote this down because we always ask for specific questions. So in your interviews with your clinician or with your research group, you should ask, what is the problem? What is the actual unmet need? What information should the test give you? Are you looking for a rule out test? Are you looking for a rule in test? Do you want a yes or a no? Do you want 3 categories, high, intermediate, low? Do you want a score? And on the basis of that outcome, what the clinician says, you ask, how would you intervene? If this patient receives a higher score, what will you do? And actually, this is the point where often the train of thought stops, because then the answer would be, oh, there's actually no alternative. Or we can do something additional, but it doesn't give you improved patient outcomes. So going through these questions, I would say would give you an advantage early on, because if a test performance is so we could improve it. If the technology platform is not relevant in clinical laboratories, we could do a bridging to a different technology. It's all difficult, but it's manageable. If your input material is not stable, we could try to do things in the workflow to make it stable. We can reduce costs, we can make it more efficient. There's so many things you can do, but when you don't have this unmet need clear, you have to reject the opportunity anyway. Because at the end of the day, unless major things change in the landscape, it will also not be applicable 5 years from now.
Yeah, I would encourage all of you to check the notes and write down those questions that Marika just listed off. Those are very important questions. I think that's a really key point. I boil it down to what do you do with a positive result? Like you have a positive result, what next? Right. And then like what happens in the clinical workflow? And you can kind of start from there. But I think she laid it out really nicely, like the different things. And that is very important for you to understand early on. So let me tell you a little bit about how I look at things currently in my current role. And I want to emphasize that different companies are going to have different strategies and going to take a slightly different perspective. So in my current role, I'm looking for things that will quantitatively improve our cancer screening performance of our existing platforms that we have internally. And so for that, I'm looking for biomarkers and prototype assays that have the potential to improve performance for, colorectal cancer detection from blood, lung cancer detection from blood, et cetera. So we meet with a lot of people who have biomarkers, and usually it goes something like this. They have, which is how I did it when I was in academia too. So no offense implied, but you have samples that are collected and processed immediately because you want the freshest, cleanest samples, and then you get the strongest, cleanest signal, and those are used for discovery. And then you take a subset of that same cohort and you use it as an independent or held out validation set, and then you get your top tier publication and you celebrate. Right. And then you turn to Iris, and then Iris comes to me and says, Dan, do you want to license us? And I say, well, we operate a central lab model, which is pretty common in the United States. And so there you need to consider sample stability, right? So the time between the blood collection and when it's processed and when you analyze the sample. So that is kind of the valley of death for a lot of biomarkers, is just that sample stability. And so that's one of the first things that I look for and that I talk to people about, is if they have any data like that or if they've thought about that at all. And then the second thing is an independent validation cohort. So yes, you did a cross-validation, or you even held out a set, and that's good for publication. But I can tell you that a lot fewer than 10% of those end up actually working down the line for various reasons. Like I mentioned, it's kind of the pre-analytical blood stability reasons. But then there's also a lot of times, I see people use these powerful machine learning approaches and get really strong results, almost perfect ROC curves. And then it ends up that the machine learning has learned like something about the collection site or some intricacy of how it was processed that you're not aware of, that it's learning, and it's keying off of that. And so then when we test it in an independent cohort of samples, it just falls flat, right? Or the performance drops considerably. And I guess that's the last thing that I will add, is at an early stage, the performance has to be quite strong, because I've looked at hundreds of things now, and I've never seen the performance improve as it progresses to clinical use, like literally never. So the performance will just, as a stepwise manner, get worse and worse until it's clinically implemented. And so when I was at Roche, part of how I viewed my job in assay development was to preserve as much of that awesomeness as I could, from like the early research stage down to the clinical implementation. Because because it's really like, oh, you've got to scale up production of the reagents. Oh, it's got to be more stable. Oh, it's got to be easier to do the workflow. And that just, the performance just erodes slowly over time. So what I look for, just to recap, is independent validation. What do I think is going to happen with the blood, the analyte stability in blood? Is it going to be compatible with the central lab model? And if all that checks out, then yes, IP, it's very important that you have the IP locked up. Otherwise, it's just a nice idea that other people will copy. And then we actually evaluate it, and we send blinded samples and we have them tested, and we compare the results to what we get from our internal platforms, and we see, oh, you can provide a 15% sensitivity boost for us in this application. So that's worth a lot to us. So let's talk, right? And then that's how we think about licensing.
Yeah, that's super helpful. And then I guess for our researchers thinking about putting together data packages as they're coming to potential partners, what is the typical data that you would like to see when someone is coming to you with a partnering opportunity in the biomarker diagnostic space?
So I would like to see how it works, right? I would like to see a peer-reviewed academic publication. I would like to see data from an independent cohort of samples. And I don't know, those are kind of the big ones. I don't know, Marika, what do you think?
Yeah, agreed. And then usually we have our bioinformaticians looking at how they did the modeling just to get a general idea of how complex or not complex they did the modeling. Because we don't do it immediately, but down the line, we should be able to reproduce the results bioinformatically too. And usually early on in the discovery paper, they can already see the models used and whether that would, yeah, it would hold in the independent validation situation.
Mhm. And then what about regulatory approval or reimbursement? Are there any red flags, or do you think about whether technologies will struggle with regulatory approval or reimbursement, or is that not a part of your calculus?
Yeah, that is a factor. So let me call out mass spec based technologies for a minute. Just as an example of things without much like established IVD, say, precedent, right? So if you're gonna, teach the FDA how to do genomics, or you're gonna like forge a new path, like, from a regulatory approval standpoint, or I mean, we just look at the example of Grail in the cancer screening space, right? They were really leading the charge and saying we're going to let the science and the market tell us, and we're not really going to follow traditional regulatory um, or reimbursement pathways, right? So generally speaking, I look for how hard it would be to integrate something into our existing platforms, and also how hard it would be to do analytical validation and to get kind of the FDA to approve such a test. So if it uses a completely foreign technology, there are no existing in vitro diagnostics based on that technology, that's going to be a harder. Then you want to look for just kind of basic analytical performance things like, is it linear? Is it like reproducible? Stuff like that, right?
Yeah, and in addition, if you look at reimbursement coverage, I think looking then at Medicare for the United States situation, you can see all these policies that they already have in place. And when there is already an existing LCD in that space, going after it, it's a bit more time efficient. So your path towards coverage is much faster than at the point that there is no LCD yet, and you have to first apply for a new LCD, have this commentary period of 12 to 18 months, and then submit your technical dossier. So evaluating opportunities from a reimbursement perspective, coverage perspective, I would say that well, if there is already one or two players ahead of you in the market, they kind of pave the way for a more efficient access to coverage.
Yeah, it's interesting. It regulatory in my view is a bit more black and white as far as, okay, if we're going this what they call laboratory developed test pathway, and we're going to build out a CLIA clinical lab and do this central lab approach and kind of offer it as a service at this single lab versus going the FDA routes and doing an IVD. Reimbursement becomes a bit more gray and almost a black box seeming challenge where it's quite uncomfortable taking an assay and trying to figure out, okay, what check marks do I need, to achieve with it. So to your point, it's it's funny because you look at competing technologies or tests and man, this is our competitor. And actually with reimbursement, it's interesting because as Marieke mentioned, if you're actually a second to market, or even if you're going for a new policy through Medicare or one of Medicare's contractors, these local coverage determination policies that are written aren't just usually for, or anymore, I think it's very rare that a billing policy is written for one single test. It's actually kind of all encompassing for that area. So if it's minimal residual disease, for instance, MRD testing for cancer patients, Medicare through this MolDX program has a local coverage determination policy for the different indications, let's say colon cancer, that helps everybody in that space. And then it's figuring out, okay, what type of data do we need to present to them? Whereas if you are charting unadventured waters in a space, well, then it might be a longer pathway. You might need to go in and actually request for a new policy, which can be a multi-year pathway. So and that's part of the importance of engaging with these different stakeholders so early to build a better understanding and better expectations, more realistic expectations of what it's going to take to get this to a product that's actually paid for.
Yeah, I guess digging into that, Daniel, I'm curious about your own personal journey, deciding which regulatory framework to pursue and what that was like.
Yeah, so for our EsoPredict test, this prognostic assay, so EsoPredict is used for these patients that have this condition called Barrett's esophagus. And this is this pre-malignant condition for esophageal adenocarcinoma. So patients are diagnosed with Barrett's esophagus endoscopically, usually by their GI physician. And once they see that this metaplastic change has occurred, they're put on this periodic endoscopic surveillance program, primarily looking for this precancerous abnormality called dysplasia. So our test uses methylation-specific PCR. It's ran through our algorithms, and we give a 5-year probability for progression. So is this patient lower risk for progressing? And in that case, we can reduce surveillance potentially, or are they at higher risk? Maybe it's more intensive endoscopic surveillance or even a treatment, usually via ablation or endoscopic treatment. One of the challenges for it, so regulatory-wise, often going through the FDA pathways, it's a pathway where you need to lock your assay, and it's often prospective clinical trials. Becomes really challenging for prognostic assays, where the most feasible studies, especially for a company, but in academia or at a company, are retrospective studies, where we already know what has happened to those patients. What are the outcomes of these patients? So pretty early on, we decided on the strategy of going what's called the CLIA pathway, or developing a laboratory developed test. And this can be a lower cost pathway. You're getting your laboratory validated and certified and licensed, rather than just taking that unique assay through, although you do need to prove out analytical validation and really clinical validation. But it can be a faster way to market. It can be a lower cost way to market to get an assay to be usable in the clinic. Now, having said that, it also comes with its different challenges. It can only be done in that one central laboratory. Reimbursement wise, if you don't have FDA clearance, it cannot, certainly with guidelines, with reimbursements, you are at a little bit different of a starting point as you go towards these things. So, so there was a few considerations. One, for the regulatory, as I mentioned, we decided to launch this as a laboratory developed test. And then reimbursement, without going too far down the rabbit hole with reimbursement, there became a number of questions as far as, okay, do we want to do this out of our own laboratory? And what area, what geography do we want our laboratory to be set up? Because it is treated different depending on what Medicare administrative contractor you fall up under. And in the United States, I forget, it's like 7 or 8 different regions. And based on the MAC, they call them, these Medicare administrative contractors, they have different ways of dealing with diagnostic tests and different pathways for getting coverage. Some will pay really early, and then it's kind of like this pay and chase where they're going to pay early, but then they're going to follow up, and they're going to try to add you to a billing policy. Some start you in a non-coverage billing policy from day one, and then from there you need to go and do a technical assessment through a current coverage policy or go and try to get a new coverage policy. So I won't go too far down the rabbit hole with that. It's the reimbursement, to me at least, is kind of the grayest world and was the most uncomfortable world jumping into, because as I started talking to people, it's like more and more questions just kind of emerged around it, and it's much less clear. And with that, it's I found the best thing to do is to find those right experts who have navigated it, and then kind of work through them to identify the best pathway for getting it to as a paid test and covered test.
Got it. Yeah, that's super helpful. I think that the regulatory landscape is something less familiar to us as academics. So I want to remind the audience now, if you have any questions, please feel free to drop them in the Q&A. I think we're going to pivot to just general lessons learned and your experiences commercializing novel biomarker technologies. So I guess in summary, looking back at your experiences, what are some of the most unexpected challenges or lessons that you faced when you were trying to transition a biomarker from discovery to a clinical product?
Who's going to go first?
Go ahead.
So, right. I think probably the things that, I would not have believed if I had like a time machine, I could go back and tell myself these things. I think the first is, it's not like that the invention is the relatively the easy part. I mean, I like I've been there, so I don't mean to be offensive, but in terms of time and investment and success for commercialization, that initial figuring it out part tends to be the shortest and least investment intensive part. So that was definitely a big lesson that I learned. The second thing is that it's usually, your invention has all these cool, really sexy applications. It's going to change the world. It's going to revolutionize clinical practice. I mean, that's how you write your fellowship proposals and all kinds of stuff too, right? But that's not what ends up being a successful business, right? I think, Daniel has a good example that he kind of mentioned previously, but like, it's those routine clinical use, the boring, the currently reimbursed, with a regulatory precedent that keeps the lights on. It's the boring stuff, the boring uses of your application, or the pivot to the less sexy cutting edge. And because ultimately that allows you to keep your revenue stream going and then build to those more cutting edge revolutionary applications of your invention. So yeah, I think those are the two things that really stuck with me. And then maybe the third one is that, like I mentioned before, the performance always gets worse, right? So it's depressing, but it its treatment.
We have a question I just want to flag while we have some time. If you've identified a novel biomarker for a drug that is already approved, will this have any commercial potential?
If the drug is approved without requiring the biomarker to be tested, then the answer is unfortunately no. I mean, yeah, other panelists can feel free to disagree with me. But generally, right, a pharma company wants the largest total addressable market possible for their drug. That gives them maximum revenue. And so if you come along with a test that maybe identifies a subset of that population who responds better, but the drug is already approved for use in a larger group, generally the pharma company will actively not want to limit their target population.
I'd throw in one potential caveat, which is, say you have a second line medication and you're competing with, I know in the spot like eosinophilic esophagitis, this inflammation condition in the esophagus. There's a number of second line drugs currently that are being FDA approved. And one of the questions becomes, okay, which drug do I move forward with? And are there biomarkers that can help us determine which drug is going to work best in this patient? Potentially, and it's an area that I don't have a ton of experience with besides some of our own discussions looking at biomarkers. But potentially there could be utility with that. And at least from my perspective, worth reaching out to some of these pharma companies or working with Iris and Chris to see if maybe you could have a discussion and see whether it's something worthwhile to pursue commercially.
Yeah, I actually agree with both of your analyses, to be honest. It really depends on the situation. What I also know is if a pharmaceutical on the market is at the end of the lifetime of their patent, doing companion diagnostics may be a reason for the patent to be extended over a longer period of time. That would be a strategy to code for. What I would also like to address, and I think it's one of the lessons that we learned, is that you always have to take the future clinical landscape into account while developing, because you're on the market 5 years from now. And we are also active in the field of multiple myeloma. It has many pharmaceuticals already on the market, but there are so many introductions every year. So the moment you have a response prediction classifier for a product, a pharmaceutical product, by the time you're on the market, it has already been substituted by a second or third generation. So if you are in a very fast-paced therapeutical landscape, predictive markers are quite difficult to commercialize. I don't want to discourage anybody, so it's very it's also very depending on the situation, of course. But yeah, try to find out how it looks like so many years from now, 3, 5, also depending on situation. And then if it's still relevant, it could have an opportunity.
Yep. I think I always say I never say no the first time. So yeah, I definitely reach out to people and have the conversation. I think the general principle is the pharma company is going to want to have a larger potential market. So if it gives them first line access, if it gives them a leg up over the competition, if it allows them to extend, patent life, all of those things would kind of give them access to market that they don't currently have access to. It would give them a better shot.
Thanks for that. I guess I want to give the other panelists an opportunity to answer the question about lessons learned, either in your experience with your own startup or working with academic inventors like you do, Marieke.
Yes, I think it's really important to get out of the research situation. And I think our very first product is also, it does so well in the research setting, but then on several aspects, it's just not ideal for the clinical setting. It's currently used, it's commercialized, it's being used in the clinic, but if you look at the time span it took to get to there, it's much longer because we were kind of stuck in the research setting in the beginning. So we could have done this faster. I'm not saying it could have been quite as fast. So indeed, you have when I see the poster public presentations on all these, like the AACR, the big new cancer research presentations, I always think, wow, there's so much possible in academia. Like, it's a really cool factor, like these technologies that can do anything. But it's also true what Dan said, that going to the clinic at the end of the day, it has to be translated to super boring, almost old-fashioned technologies, to be able to work in a clinical laboratory. So making that transition early on, and also ensure that you do your how do you say, your improvements on your product with that in mind, I think it's really important.
And for me, you know, starting this company, I started it with my faculty co-founder, and it's been one of the most challenging and uncomfortable journeys, kind of going through all the pieces we talked through today, and the oh my goodness, now this and another area we don't know about, or it's going to take this long, it seems to do this, and man, it's like we used to sit back and think about, man, imagine if one day this is used by people, and if this works, and one of the most rewarding experiences I get to live now is, I mean, we were sitting in an office at a lab talking about, man, maybe we could do something with this, and really being totally unqualified to take it. Not, I mean, just not knowing so much. And today we get to have discussions with physicians who are using our tests and see orders coming in every day, and hear these personal stories about how we were able to use these test results and we're able to get important treatment to this patient earlier. Or, hey, we were able to use this to reduce this patient's anxiety. They had real concerns about this condition and this unique individual story. And so it's a challenging journey, but it's also been just the most rewarding journey of being able to watch this early technology, crude, with little understanding on our parts for how to do this, but with some willingness and open-mindedness and being moldable by others, and kind of flexible in our journey to pivot when needed. It is just one of the most awesome things. So definitely would encourage anybody with interest out there. It's stay hungry and stay persistent, and it is possible to take that technology and to see, whether it's that technology or that version of that technology, it's possible to take something from academia and to go out and really make an impact with it.
Well, great. What a lovely message for our researchers, and a great way to end the panel. So with that, I think we're just about at time. I want to thank the panelists for your time and insights, and all of our attendees for joining. We host lots of programming through Weill Cornell Medicine Enterprise Innovations, so please check out our events calendar if you're interested in similar events. If you have certain topics which you're interested in hearing about next, please let us know. I hope you all enjoyed the panel and came away with a better understanding of the different opportunities and challenges specific to this space, or even an idea or some drive to commercialize your own biomarker discovery. And with that, thank you and have a good day. Thanks all.
Thanks.
Thank you. Bye.